All right, Megan, if you're ready, I'll get us started. So first of all, welcome everyone. Thank you for being with us today for our Research Rising Lifespan Institute seminar. I am delighted to welcome Dr. Meghan Puglia as our speaker today. Dr. Puglia is an assistant professor in the pediatric division of the Department of Neurology at the University of Virginia. She completed her doctoral training in cognitive psychology at the University of Virginia, where she studied infant brain developments related to social behavior. This work impressively was supported by multiple fellowships, including an American Psychological Foundation Kopitz Award, and then an NICHD F31 NRSA Award. Prior to her time at UVA, Dr. Puglia completed her BS in Psychology and Neuroscience, graduating cum laude at Penn State University. She moved on to the faculty at UVA immediately upon completing her PhD and has been highly productive in her faculty career. She served on or is currently serving as an investigator on multiple extramural funding awards from the NSF and from NIH, including serving as a co-investigator on a current NIH ACE Network Award focused on biomarkers of late diagnosis in female and diverse autistic individuals. This is an ACE that's been around for a number of years. Actually, it's been really productive, really tackling a field that was untapped in many ways prior to this work. Her current efforts also are supported by an NIMH K01 award currently, investigating neural variability as an early emerging biomarker of autism. I think she's going to be talking to us about a lot of that work today. Her studies consistently have been published in highly rigorous and high impact neuroscience journals, such as Developmental Cognitive Neuroscience and BMC Medicine. And I had to note that she published a first author paper in PNAS, which itself is an accomplishment, but it was actually the #1 most read journal article in the year that it was published, which is just really cool. Her current position is fully research dedicated, but it's actually important to also notice that even in this context, she has actively sought out teaching and mentorship involvement, including development of a new course that was over-enrolled. She's earned an excellence in mentorship recognition from UVA, and she often includes students as co-authors on her publications. Her work is innovative, it's integrative. She uses cutting-edge systems neuroscience, behavioral epigenetics, and data science approaches to understand basic questions about individual trajectories of early social development and their neurobiological substrates. Today, she's going to be talking about using these approaches to advance earlier detection of autism. So Dr. Puglia, welcome. Take it away.
Thank you so much, Matt, for that introduction, and thank you all for being here. It's been such a pleasure to virtually visit. I wish I could be there in person, but it's been really great to get to speak with many of you, and I look forward to speaking with many more tomorrow. So yeah, today I'll be speaking about some of my efforts to help us move up the diagnostic age for autism. So I'll launch in with first an introduction to my research program in general. So I really take a gene-brain behavior lifespan approach. I've published everything from infants to elderly adults aged 80 and above. But the primary focus of most of my research and the work I'm going to talk about today is on a specific gene, the oxytocin receptor gene. And within the neural realm, looking at a very specific marker, trying to understand something that traditionally we would throw out of our analysis as neuroscientists, which is the random fluctuations or seemingly random fluctuations, or what I'm shorthand referring to as neural noise, and I'll describe what I mean by that. And then at the behavioral level, I'm really interested in social signal detection and attention to social signals. And the reason I choose to focus on this is that I really see it as kind of the first stepping stone for a person to be able to engage socially. So before you can decide, is this face that's approaching me angry or happy? What's the intention of this person? Can I understand what they're trying to mentalize? You really have to first be able to detect that there's a social partner there. So how is this ability set up, and how does it vary across individuals? And how can we capitalize on these gene-brain behavior interactions to really understand what interventions might benefit each individual person at a particular time in their lifespan? So why should we even care about social functioning? Obviously, there's strong implications for developmental disorders like autism, which many of you are interested in. But even more broadly than that, this is something that's really important to us as humans. We're innately social beings. This is one of my most favorite studies to highlight here. So this was a meta-analysis of over 148 studies, over 300,000 participants, looking at basically what causes a healthy life, what helps with longevity, what might help us live longer. So the way to read this is the further along the graph here that the chart will go is showing us increased odds of mortality. So they looked at all kinds of factors. So we know, for example, certainly environmental factors can impact longevity and health. And certainly they found that individuals who live in a location with higher air pollution compared to lower air pollution did have increased odds of mortality. We also know health factors certainly are very, very important. So for example, individuals with lower physical activity or an obese BMI certainly had detrimental health outcomes and decreased longevity. Behavioral choices certainly as well. So for individuals who were excessive drinkers or smokers, very significant increased odds of mortality. But what this study found was absolutely remarkable to me. Akin to smoking 15 cigarettes a day, individuals who have low social support or feel lowly integrated in their social networks had remarkably increased odds of increased mortality. So again, that's on par with, if not stronger than, smoking 15 cigarettes a day. So we're innately social beings. This is highly important to all of us, no matter where we fall on a spectrum or what our neurodevelopmental status is. So hopefully this convinces you that understanding what kind of neurobiological factors really drive our social behavior and enable us to form these critical social relationships is something that all of us should be interested in and care about. So what cognitive and neurobiological factors do support our ability to form social relationships? Well, these begin very early in life. Attention to social information, as I said, is really my primary focus of interest. It's the first thing that allows us to, be able to learn from social abilities, detect social partners, including our very early caregivers. And this ability does emerge very early in development. So already within just the first few hours of life, these neonates are preferring to look at and attend to face-length stimuli. So whether that is kind of like a little very low level, eyes oriented up versus inverted, the baby spends significantly more time looking at that, or whether it's an actual face that is oriented correctly versus inverted, babies spend a significantly greater time looking at that face oriented stimulus. So there's a perceptual bias for social stimuli that emerges very, very early within the first few hours of life. But we do know that this ability to attend to social information or preference to attend to social information does vary across individuals. So one factor that might have this vary is diagnosis of autism spectrum disorder. So individuals with autism here are plotted in orange, and this is them viewing a very social scene. They're watching a movie where there's people interacting, and we can see that the orange traces here show that the individuals with autism don't spend the same amount of time looking at the faces. They might look at the mouth region a little bit, but they tend to look much more at background information or other parts of the scene, whereas unaffected individuals plotted in yellow really do spend a significant amount of time looking back and forth between the faces and particularly the eye regions of the scene. This is not just limited to individuals with autism, though. Certainly across the entire spectrum, there's differences in how individuals attend to social information. And this ability to attend to social information, or perhaps bias to attend to social information, is also under genetic control. So this was a really cool study that looked at non-siblings, so completely unrelated individuals, dizygotic twin pairs who share 50% of their genetic makeup, and monozygotic twin pairs. And they had them undergo an eye tracking task similar to what you would see in the previous image, where we can see what part of the stimulus are they looking at. And here we're plotting the amount of time that one individual looked at the eyes compared to another individual. So for folks who are not related, there's essentially no correlation. So however one person looked at the eye region doesn't really impact how the other person was looking at the eye region. For dizygotic twins, who again share 50% of their genetic makeup, there is somewhat correlated. So the way that your sibling looks at the eye region is somewhat predictive of how you might look at the eye region. But for monozygotic twins who share 100% of their genetic, this was almost a perfect correlation. You don't see data like this typically. So this was really remarkable. So if one twin was going to focus on the eye region, the other twin was highly likely to focus on the eye region. If one twin was not likely to look at the eye region, the other twin was also very much not likely to look at the eye region. So there is some kind of genetic factor here. So what my research is interested in trying to understand what kind of biological factors, what biological systems might be driving this effect. And an obvious target when we want to understand social behavior is, of course, the oxytocin system. So this is very classically understood to be important for regulating social behavior. So for example, oxytocin's related to things like parenting behaviors, mate selection, and also things like attention to the eye region. And taken together, all of these different behaviors that oxytocin might impact, it could be suggested that oxytocin might be enhancing the salience, or the ease with which someone can identify and detect and attend to social information. So for example, perhaps Dan becomes more receptive to the distress cries of her pup. Perhaps a mate is better able to detect whether there are signals of fitness in a potential mate, or perhaps the eye region is becoming particularly salient and contrast heavy for someone trying to detect a social partner in a complex scene. So my research is interested in testing this social salience hypothesis of oxytocin and really trying to understand what is the mechanism by which oxytocin might be boosting the ability for us to detect and attend to social information. So the actions of oxytocin in our body is a naturally occurring mammalian hormone. All mammals have it, but the actions are dependent upon its receptor. So that's this molecule here in blue. And its receptor is coded for by just a single gene, which is very aptly named the oxytocin receptor gene, or OXTR. And this gene undergoes an epigenetic modification called DNA methylation. So epigenetics, epi meaning above the gene, are an interesting model for us to use. So unlike genetic changes where there might be a single nucleotide polymorphism or a copy number variant, where the actual genetic sequence itself can change across individuals, in an epigenetic modification, the two different statuses of a gene can be absolutely identical as far as their nucleotide sequence. But what occurs is a methyl group binds to the cytosine, and that actually constricts the chromatin so that transcription factors can't come in and bind, and that gene can't be transcribed and used by the body. So again, these are two identical sequences of genes, but all that occurs is that a methyl group binds to a cytosine, and that can occur when a cytosine and a guanine are adjacent to each other, constricts the DNA, and makes it harder for it to be transcribed. And so OXTR, like all genes, can undergo this epigenetic modification, DNA methylation. And we've found a very specific site, a couple of sites that are functional. So not all DNA methylation can actually regulate gene expression. So for example, some of us might have sites where all of us are very highly methylated, or all of us are very lowly methylated. It's not actually functional or variable across individuals. So very, very critical first step is to identify what sites are actually functional, which sites should we care about. And of course, for human behavior, the causal tissue of interest is as the brain. So methylation values can vary across tissues. So as you know, hopefully all of us have the exact same genetic sequence in all of our cells, but one cell might become a liver cell, one cell might become a hair cell, and this is due to epigenetic factors turning up and down different genes so that the genes are expressed in different ways in these different cells. So for human behavior, the brain is the causal tissue for us. So very, very critically, some work that my graduate advisor, Jeff Connolly, did to really pioneer this work and set the stage for us was identify sites in which, in the promoter region of OXTR, that methylation and gene expression were actually correlated. So that actually means that as methylation was increasing, gene expression was going down in the brain. So those are regulatory sites that are functional. So in short, as we have increased oxytocin receptor methylation at these sites, there's decreased expression of the receptor, so presumed decreased ability to use this important molecule. There's also really a strong interest in epigenetics for early life experiences, because these markers can be turned up and down, so they're kind of like a dimmer switch, and in particular, very early life experiences can impact how these methylation markers are being put down. So it's really important to understand how these markers are reflecting neurodevelopmental differences. And Also, of course, that initial study was done in brain tissue, which is very important. But if we want to be studying healthy babies, I don't think I'd get any family to sign up, nor would I ethically want to if it required, doing a biopsy on a baby's brain or any participant's brain that did not need that for medical reasons. So we need a peripheral tissue that's accessible and a proxy for what's going on in the brain. So to identify this, we turned to a prairie vole model, which has a highly conserved region of the oxytocin receptor gene. So there you have a lot of overlap with the way that their gene is set up. And they're also highly social phenotypically, more so than other rodent models. So in collaborative work that I've done with prairie voles, we were able to establish that early life experiences, in this case, prairie vole pups that received a lower or a higher level of care would change the level of methylation in both the brain and the blood of these pups. So for vole pups that have higher levels of care, we actually see decreased levels of gene expression, or methylation in both the brain and the blood. And again, decreased methylation is increased expression of the receptor, increased ability to use oxytocin. So both of these tissues in our prairie vole model are responding to developmental experiences in the same way. And similarly in humans, so this was a group of individuals with autism and neurotypically developing controls. And again, in the brain, we find individuals with autism who we know have a different developmental experience with social behavior, social stimuli, and social perception, had increased levels of methylation in both the brain and the blood. So again, increased methylation, decreased expression of the gene, perhaps decreased ability to use endogenous oxytocin. So this was really important work to establish this kind of early life experiences might impact in both the causal and the peripheral tissue, how our body is being set up for processing oxytocin. And then finally, certainly wouldn't want to take blood tissue, or brain tissue, but even blood draws can be not ideal for not just infants, but also individuals who might be more sensitive to this. So some of the first work I had to do was establish that we can also reliably assess our sites of interest within the saliva, and indeed we can. So here I'm showing you plotted along the x-axis, methylation levels for about 140, 180, depending on the tissue type, levels in the blood and in the saliva along the y-axis for the same individuals. And we can see that these are highly correlated. So what this work has shown is that we can use the methylation levels in the blood to be a proxy for what's going on in the brain, and then the methylation levels in the saliva to be a proxy for what's going on in the blood. So again, particularly because epigenetic markers are very tissue specific, this was really powerful and important work that had to be done before we could go on to see how are these levels perhaps reporting on what's going on in the brain as our individuals are developing. So the hypothesis that I have been interested in testing and working on is that oxytocin might be increasing the salience of social signals by boosting this neural noise in response to social information. And in particular, increased access to endogenous oxytocin very early in life might be increasing this neural noise and positively impacting social development. So these first series of studies that I'm going to tell you about are testing this hypothesis. But first I want to describe what do I mean by neural noise. And it might seem counterintuitive that I'm saying increased noise is actually going to help signal detection. So here is a little bit of an illustration on how that can be a possibility. And this is a phenomenon called stochastic resonance. And the idea here is that if you have a subthreshold signal, so say, for example, this is one neuron that's trying to talk to the next neuron, and the detection threshold for the signal to be propagated to the next neuron is this dashed line here. And so here, this is subthreshold, so it's not quite reaching its next target. But if you add a moderate amount of random noise to the signal, it can actually boost the fidelity of the signal so that it's accurately represented. So here, now every time that neuron is trying to talk to its partner, the output is being received. There is likely a sweet spot, however, so too little noise or too much noise, and again, the signal is not being well propagated. So what we anticipate is that there's likely to be, an inverted U-shaped curve where there's an optimal amount of noise that is increasing signal detection. And this type of work has been shown to be the way that the brain is processing. So for example, a moderate amount of variability in brain signaling does facilitate the exchange of information between neurons. It enhances neural synchrony, so neurons that are firing together or wiring together, meaning that it's setting up dynamic, robust, adaptable networks. And also a way to think about the benefits of noise are, for example, say you take the exact same path to work every day, and then one day there's construction and you can't drive on that path. You have no idea what to do. You've never taken a different path. Same kind of idea where you really want to have variability as networks are being set up so they can really explore their full state space, so that if a node is knocked out, like in this lower image here, there's already been different paths explored and the signal can propagate, and these networks are really robust and dynamic. So yes, it also increases the dynamic range. So all of this has already been shown to be true for a little bit of variability in the brain. So it's increasingly understood to have a very important functional role for us. And very interestingly, oxytocin, in addition to being a hormone, is also a neuromodulator, and it has specifically been shown to impact things like signaling and neural variability in the brain. So for example, it enhances signal to noise ratio, it balances excitation and inhibition, which we know leads to optimal levels of neural noise, and it increases neural synchrony. So oxytocin's actions in the brain might specifically be driving the appropriate level of noise, and perhaps specifically in response to social stimuli. So we've seen that neural noise is an important functional part of brain systems, but how do we actually quantify this? Here I'm showing you 3 simulated time series. One is completely random noise, the top one up here. The next one here is a sine wave, so it's completely deterministic. At any one point, you know what's coming next. And then also this is some EEG data on the bottom here from one of our babies, which is a bit more complex. Now, all of these signals have the exact same number of data points. They have the exact same mean. So in our traditional mean-based analyses, where we kind of like average across different trials or different subjects, we wouldn't be catching any difference here because these have the same mean and they even have the same standard deviation. But just by looking at these signals, we can tell they're very, very different. So how can we actually quantify that? One metric that has been really emerging in popularity and that I've been primarily focusing on is something called sample entropy, or entropy. So this is a measure that quantifies the unpredictability in a time series. So basically it's just looking for patterns that repeat in a signal. So a completely random signal where at any one point what's come before does not at all inform you about what's coming next, that would have a high entropy. So white noise will have a high entropy level. A completely deterministic signal will have low entropy. You always know what's coming next. The patterns are very repetitive. But what about a really truly complex signal, like a biological signal such as heart rate, or in our case of interest, EEG signal? So for this, we really need to understand the fact that these systems are operating on multiple timescales. So as the different regions of the brain are interacting, different synapses are coming together, forming up to local networks, all the ways up to large-scale networks. There is noise that's fundamental at each level of these, each of these levels in the hierarchy of the brain. And as that kind of propagates, we need to consider the fact that the brain is a multi-scale system and understand how this noise is operating on different timescales. So for this, we turn to something called multi-scale entropy. And basically, all that is you take your original time series at the level that you recorded it, and you just coarse grain it. So basically, all you're doing is zooming out to increasingly coarse-grained or slower Hertz scales, for example. And it's been shown that the really fine scales closer to the sampling rate, the original sampling rate, are involved with local information processing, whereas you kind of zoom out more and more into those more coarse-grained timescales. We're getting more information on the long-range integration of information across disparate brain regions. And this type of multi-scale entropy analysis has been looked at cross-sectionally. And what we've seen is that individuals, as they age from early adolescence into kind of prime cognitive young adulthood function, we see an increase in entropy across pretty much all timescales. So here we're going from adolescence up to adulthood, we're seeing an increase in entropy. And what we're also seeing is that As entropy increases, these individuals who are more entropic tend to perform better on a task, so higher accuracy and faster reaction time. So again, this is highlighting that at the right level, a moderate amount of variability or unpredictability in brain signaling is a good thing. So for my NRSA study, I was interested in seeing how oxytocin might be influencing brain signal entropy during social perception in a neurotypically developing population. So this was infants that were enrolling in the study at 4 months of age. They underwent EEG and some behavioral measures and provided a DNA sample for exploration of oxytocin receptor gene methylation. And we were particularly interested in just understanding normative variability. So these were all babies who were born full term and did not have a family history of a neurodevelopmental disorder. So for the EEG paradigm, we explored both social and non-social perception within the visual and the auditory domains. So within the visual domain, the babies watched videos of women who would turn their heads and smile. Video is lagging a little bit. Hopefully it's coming through on your end. And then for the non-social control condition, it was just fruits or vegetables, common objects that would rotate. And then on the auditory domain, the babies within the social condition would listen to motherese speech. So this was recordings that were actually made as mothers were speaking to their babies in the home. Let's see if we can.
I'm going circle. Heart.
And then as the non-social control condition, it was sounds of water, so things like this. And very critically, both the social and non-social stimuli, so in the visual domain, these stimuli were matched on luminance, spatial frequency, contrast, and visual angle. So the only differentiating thing was whether it was a social stimulus or a non-social stimulus. And similarly, our auditory stimuli were matched on mean fundamental frequency, duration, and intensity. And the water sounds were specifically selected to be something that the baby would also have a high amount of familiarity with. And then to assess the baby's visual and verbal social behaviors, we had them undergo an eye tracking task to see their attention to social information. So here, if this video will start. We have a baby undergoing an eye tracking task. So mom wears sunglasses so the eye tracker won't pick up on her eyes by accident. And so everywhere the baby is looking is the red point. What the baby does not see, they don't see the red point, and they also don't see these outlines here. And I'm not sure if the video is lagging for you guys as well, but basically what we can see and quantify is how much time does the baby spend looking at faces compared to background objects or the hands, other non-social information in the video. So again, the white lines are just illustrating for us how we're defining our areas of interest. The baby would just see the video without that additional information. And sorry that it seems to not be loading. But we also were interested in assessing their behavioral or their verbal behavior. So for this, we looked at the short infant behavioral questionnaire, which has a vocalization domain. And so we can ask how often was the baby verbalizing in different types of situations. So for example, how often did they squeal or shout? How often did they coo when they were being undressed? Again, these are 4-month-olds. Trying to get at how much vocalization they're making in their age-appropriate domain. We also did have adults undergo this paradigm. So the stimuli were identical, except we didn't use the motherese because it felt just a little bit condescending, but it's very similar, just two people speaking single utterance words for the auditory social stimuli. And then to assess the adult's behavior within the visual domain, they underwent a biological motion perceptual threshold task. So if it were to play, you would see highlighted in red. And again, this is just for illustrative purposes. The actual task would be all the points are in black. There's a point light display of biological motion. So for example, someone throwing a ball where we're only capturing the actions of the major joints. And and I guess in the freeze frame at least, you can see how there's additional points being added. So they see two displays back to back, and they're asked to do a forced choice. Did the first display or the second display have that biological motion? If they get it correct, the algorithm adds more noise dots, so it's harder to pick out that biological motion. And if they get it incorrect, it takes away noise dots. And then the algorithm continues until it converges on the adult performing at 75% chance or better. And then similarly, we assessed the adults' verbal social abilities. So here we looked at their pragmatic language skills using two autistic-like trait questionnaires, the Autism Spectrum Quotient Questionnaire and the broader Autism Phenotype Questionnaire. And so this asks them on their self-report on their subtle social communication abilities. So for example, are they often the last person to understand the point of a joke, or do they enjoy or not enjoy social chit chat? And so to put together our gene brain behavior, So looking at how the brain signal entropy responded. So when we look at the group level, here I'm plotting brain signal entropy for 4-month-olds in the auditory and the visual domain. Within the social condition, it's plotted in pink, and the non-social condition is plotted in blue. And we can see that looking at the group level, there's really not a lot of difference. You can barely even see that there's a social line here, and it's overlapping so much. So the babies, on average, are not really distinguishing across these two types of stimuli. However, when we start looking at the individuals, we do see some significant differences. So some of the babies are more entropic, more variable to the social condition relative to the non-social. Some babies, like the group average, don't really distinguish across the two conditions. And some babies are actually more variable, more entropic to the non-social condition. So what we can do is take a different score to really understand relative to the social, how are these babies responding. So babies who are more entropic to the social condition will have a positive value. We did social minus non-social. And so to put together our gene brain behavior variables, we took a partial least squares path modeling approach, which allows us to look at associations, complex associations between latent variables. So basically we could take all of our electrodes and have it create an auditory latent variable, all of the scores on the different items within the verbal or visual task and our different replicates or sites of our oxytocin receptor gene, and look for complex interactions between how our gene is influencing our brain and influencing our behavior. So within our 4-month-olds, what we found was within the auditory domain, babies who have higher levels of OXTR methylation, so again, presumed decreased ability to use oxytocin, showed a negative association with their brain signal entropy within the auditory domain. So these babies were showing less variability to social relative to non-social auditory stimuli. And those babies were the same that went on to vocalize less themselves. This effect was specific for our 4-month-olds within the auditory domain. We don't see anything coming out at all within the visual domain at 4 months of age. Interestingly, in our adults, however, so first of all, we see, again, a negative effect between OXTR methylation and, in this case, behavior rather than going through the brain, modulating through the brain. There's a direct effect. I'm happy to discuss some reasons why that might be the case. But more interestingly, what we're interested in seeing here is domain-specific effects between the amount of brain signal entropy in response to the different social stimuli and the behavior. And there's remarkable specificity here. So for adults who are more entropic during the auditory perception, those same adults would report better verbal behaviors. However, there was no interaction between the auditory and how well they were able to detect the biological motion. And similarly, in the visual domain, adults who are more entropic to the visual stimuli, so watching those faces relative to those objects, those adults were able to detect that biological motion within significantly more extra noise dots compared to individuals who couldn't. And again, that is a specific path where visual is impacting visual behavior and there's no effects on the verbal behavior. So some really interesting domain specificity, and also perhaps a developmental trajectory here where infants are showing auditory effects and adults are showing both auditory and visual. So why might we not be seeing these effects in the visual domain for our 4-month-old infants? Well, the visual sensory system is one of the last to mature. If this would play, what we would be seeing is that as newborns age into the first few months of life, they gain both contrast, visual acuity, and color perception. And actually, the visual system isn't fully matured until 3 years of age. So certainly by 4 months, these infants just might not have the same experience attending to social information visually. So they might not yet have that same path between their visual brain function and their visual behavior. And indeed, so the visual system is one of the last to mature, certainly after the auditory system, which occurs even after the somatosensory or touch system. So as we're transitioning into even earlier recruitment of babies who are quite young neonates, we're interested in now transitioning from just visual and auditory into also including somatosensory and touch into our models. And that is exactly what we're doing now with my K01. So we're moving earlier into development. So we had originally started recruiting at 4 months of age. Now we're starting to recruit from as close to birth as we possibly can. So day 0, we start trying to enroll the babies. We also are now expanding from looking at just a purely neurotypically developing population to a higher risk population. So we're starting to recruit preterm babies. And we're incorporating, like I said, additional measures like touch. And this is some work in collaboration with both neonatologists and a great team of autism researchers, because the hope is to understand how these markers might be leading to risk for autism. So what is preterm birth? Preterm birth is birth that occurs prior to 37 weeks gestation. And this is a number that is continuously increasing, but currently it affects 1 in 10 births in the United States. So it is a pretty highly prevalent condition. And there has been amazing medical advances that have been allowing us to keep these babies that are born earlier and earlier in gestation alive and have better medical outcomes. So for example, 25 years ago, a baby that was born at 24 weeks gestation, which is this little point here, that's almost 4 months early, had only a 10% survival rate. But now that is increased to above a 60% survival rate. So there has been remarkable advances in what medical doctors can do to keep these babies alive and improve their health outcomes. However, there is still a lot of work that needs to be done to understand how we can optimize their long-term neurodevelopmental outcomes. So they're being kept alive, but these babies are experiencing a lot of problems, including, for example, an increased risk for autism. So that same baby who was born at 24 weeks gestation has about a 4-fold increased risk of developing autism. And this likely occurs because these babies who are born early are coming out into the world when the brain is undergoing most of its development. So the majority of prenatal brain development does occur in the 3rd trimester. And typically that will occur in a very filtered, protected womb environment. But when these babies are being born early, they're undergoing all of these very important neurodevelopmental processes without the protection of the filtering uterus. So for my K01, the study is aiming to compare and understand how preterm birth is impacting social development and neural function. So for this study, we have both a full-term arm, so that's babies who are born full-term, again, without a family history of neurodevelopmental disorders. And then we also have an arm that's recruiting babies who are born preterm. The target N for both of these groups is about 360. And we launched collection 2 years ago, and data collection has been going quite well. So we've enrolled 191 full-term babies and 169 preterm babies. We sample our full-term babies a bit more frequently. So we bring them in for the first visit within the first 4 months of life. So ideally we capture them as close to birth as possible. And then we bring them in every 4 months through age 16 months. And then we bring them back for a final follow-up visit at 24 months. where we can do diagnostic and cognitive testing. For our preterm babies, so we're enrolling these babies in the NICU. The Virginia UVA NICU is one of the largest and most comprehensive NICUs in the state of Virginia. So we're receiving babies from really all across the Southeast. And so for them to return as frequently is a bit of a challenge for them. So we are currently just recruiting at 2 time points: an initial visit where we test them ideally while they're still receiving care in the NICU, again, between 0 and 4 months of age. So as soon as they are deemed healthy enough by our team of neonatologists, we will go in and test them. And then we're asking them to come back at 24 months corrected age. So at each of these visits, the babies undergo EEG. For the very first visit, they're still really quiet and sleepy and tired, and they don't typically open their eyes that well. So we're doing a tactile condition, which I'll show in a second, and that same auditory where they're listening to speech and water sounds. And then for all the other visits, we also do add in that visual faces and objects condition as well. And then at all of our visits, we get a host of parental questionnaires to understand how the baby's developing. We're able to go into their birth records and see, were there any particular interventions or perhaps birth complications. Particularly for our preterm babies, it's really important to understand their health and how that might be impacting or confounding perhaps their risk for autism. So we're very careful at trying to understand and pull out factors that might be additionally driving risk for autism. For example, hypoxia. And then all of our babies and parents provide also DNA samples at the visits. So we launched this study 2 years ago, so we're now just starting to age into this 24-month visit, which I'm really excited. We've had our first couple babies come back, so we're finally getting to start to see some of the outcomes, and we'll be able to look back and see if any of our earlier collection points can predict which babies are having more optimal or suboptimal outcomes. So the tactile condition that we've added in is specifically designed to activate C-tactile afferents. And this is a specific type of mechanoreceptor that is really related with social touch. A very specific rate, if you stroke hairy skin cells at about 3 centimeters per second, it has a profound effect on calming and reducing autonomic system function, and really is actually how caregivers will naturally try to soothe a social partner. So for example, if a baby's crying, mothers will kind of naturally stroke at 3 centimeters per second, and it lowers heart rate, it improves breathing rate. So what we're doing is stroking with a brush at 3 centimeters per second, either directly on skin to mimic this social condition, or because C-tactile afferents are activated by the mechanoreceptor of hairy skin cells, by putting a piece of plastic that actually blocks the ability of the hair to respond, and is our non-social proxy, so that we can stroke at the exact same spot on the body at the exact same rate, and just really block the actions of that mechanoreceptor. So what are we finding with our comparison between our preterm and our full-term babies? So here and throughout, our preterm babies are going to be plotted in blue, and our full-term babies are going to be plotted in black or gray. And so what we're finding is both within the tactile, the graph here on the top, and the auditory, the graphs here on the bottom, conditions, our preterm babies are showing decreased levels of brain signal entropy, both to the social and the non-social conditions. So again, we typically think of as high levels of entropy as a positive thing. So here, as expected, we're seeing lower levels of brain signal entropy to all of our stimuli in our preterm babies. But there does seem to be a really interesting developmental trajectory going on across these 2 different domains. So here in our tactile, what I'm doing is plotting the chronological age of the baby. So we enroll within the first 4 months of life. So we have some babies that enroll within the 1st month, the 2nd month, the 3rd month, or the 4th month of life. And we can see that within the tactile domain, as we look at the chronological age of this baby, so the more time they've had experience, having tactile input, we actually see our preterm babies kind of catch up to our full-term babies. So perhaps having experience within the social domain, or within the tactile domain, excuse me, is improving the baby's ability to process and respond to the social information in the tactile domain. Now, very interestingly, in the auditory domain, we're actually seeing the opposite effect, whereas our preterm babies seem to be getting kind of worse and having a bigger divergence from our full-term babies as their experience goes on. So this is something I'm really excited to further probe and really understand how experience is also environmental influences are kind of getting into the brain in the way that the baby's biology is allowing them to process the stimuli. And this is actually a focus of an R01 application that I just submitted, where we're really interested in understanding how sensory experiences in the inter or extrauterine environment might be impacting the way these babies are processing social stimuli across different sensory domains. So as we said, the preterm baby is supposed to be experiencing sensory information within a filtered uterus. So the uterus does act as like a low frequency filter for auditory stimuli, for example. So they'll be able to hear kind of low level stimuli, but not high pitched electronic noise, for example. And obviously, of course, the tactile experiences that they get are very different as well. So how is this precocious extrauterine exposure really shaping the baby's brain development? So we've done a little bit of work to pilot and see how we can measure these things. So we have been employing Lena devices, this little purple box pictured here with one of our babies in the NICU. And this is marketed as a speech pedometer, essentially. So it continuously records all of the auditory input, and it automatically classifies into different groups what type of information the baby is receiving. So as we've been piloting this in the NICU, we're seeing that there's a lot of variability in what these babies are being exposed to. So some of it has to do with which bed they're located in, how noisy is their neighbor? Are they next to a loud crier? Some of the rooms in our NICU are more private, and a lot are also kind of packed in with lots of babies next to each other. So what we're seeing, each of these points is a different baby. There's a pretty big variety in how much speech the baby is receiving, how much electronic noise the baby is receiving, and how much silence the baby is receiving. So there's been some early work to kind of perhaps deprive babies entirely of different sensory input as they might have in the womb, but that's also probably not a really good thing. You need the right kind at the right time and the right amount. So it certainly is it's a complex thing, but having nothing is also just as bad. And really hot off the press, I am advisor to a pediatric fellow, Kelsey Sullivan, and we are piloting a reading intervention where she has parents come into the NICU and read to very, very, very preterm babies who are born prior to 32 weeks gestation. And she's using these Lena devices to record the amount of time the parent is speaking. And what we just found, so my lab generated all the epigenetic data, is for every 1,000 words that the parents spoke to the baby, there is about a 1 to 2% decrease in methylation values for that baby over time. So this occurs in both the site that I've kind of been focusing on and one that's just 10 base pairs upstream from it. So very similar effects and in the direction expected. So again, as methylation levels are decreasing, the oxytocin receptor availability is increasing. So some really promising potential for intervention work here. Also really interested in this idea that touch might be a particularly important early emerging biomarker. So what we're proposing to do in the R01 application is understand how both positive and negative. So here I'm showing you one of our babies who was enrolled in the NICU. We can see their little avatar, like all of the painful procedures that they're getting, essentially. So this baby had 3 central lines. It had a breathing apparatus and also a feeding apparatus. So every time that those lines are inserted or removed, every time the baby got a heel prick or a blood draw. So we can go into their medical records and look over time at the number of painful procedures they're getting. So those are plotted in red. The number of interventions the baby is getting. So that's things like perhaps opioids or other analgesics or therapeutic touch, massage, things like that. So we can plot over time and then look at the net pain as well. So we see a baby, this baby spent about 60 days in the NICU, and their pain, their net pain did kind of start to converge down to about a 0. And we can also look at their health score. So, are these babies needing a lot of interventions and surgeries, having hypoxic events, things like that. And again, as they get closer to discharge, we're converging on a healthy baby finally. So we can look at both their painful procedures, but then also the amount of time the baby's receiving touch, which we know is a very important component for particularly preterm babies. So a lot of NICUs and hospitals are starting to incorporate what is sometimes referred to as kangaroo care or skin-to-skin contact, where the babies are really receiving a lot of physical contact with their caregivers very early in life, and that can decrease their hospital stay, improve their autonomic function, and really just improve their health. So we're also collecting some resting state data on our NICU babies currently, and we also, again, have a non-social and a social condition. So in the non-social condition, the babies are swaddled as they typically are and just resting in their bassinet. And in the held condition, they're held either by their caregiver or a research confederate if the caregiver can't be present. And we're not presenting them with any other additional stimulation. So just rest in the bassinet for 5 minutes or rest in arms for 5 minutes held. And we're finding a really remarkable effect here. So again, our preterm babies are plotted in blue and our full-term babies are plotted in black. And what we're finding is, again, very similar to what I showed you with our kind of tactile presentation stimulus, is that our preterm babies are significantly lower in their brain signal entropy when resting in the bassinet. But really remarkably, while these babies are being held, their brain signal entropy is basically being rescued within the frequencies of their EEG data that are most predominant for this age grade. So there is a really powerful effect of touch. This is just an effect in 5 minutes. So we're really interested in continuing to explore the mechanisms for why touch might be so important and how it can be used as an intervention. So some future directions. So we are collecting DNA on all of our babies. And something that I'm really interested in understanding is how an epigenetic maturity might prepare a baby to be receptive to sensory information. So if you're not familiar with the concept of epigenetic age, basically we all have a chronological age. Of course, that's how many years we've had on life, but we also have a biological age. So I'm sure you all know some 60-year-olds who maybe run marathons and can stay up all night or whatever. It might seem like they are 40. Or you also might know some 60-year-olds who maybe have some physical limitations and just seem really slow, and you might expect by looking at them that they're closer to 70. So what kind of biological predictors are predicting how close to death basically someone is, or how mature they are relative to their age? So you can be right on your chronological age, aging as you should biologically. You can have accelerated, so you might be aging faster than maybe you should, or slower than maybe you should. So really interested in understanding, particularly very, very early in life, perhaps having a more mature epigenetic status allows you to be more receptive to auditory stimuli, and we can maybe really predict which babies would benefit from which intervention at what time. Another future direction, so the R01 application is shifting more towards MRI approaches. So most of the work all the work I've showed you so far today has been focusing on EEG, which is a really powerful marker for the temporal dynamic kind of work that I've been interested in doing, because it has much more temporal precision. You can get a lot of measurements really fast. But we're also really interested in understanding how these early sensory experiences and biological markers are organizing neural circuitry to support these social behaviors and sensory processing abilities. So we've been piloting collecting MRI data in neonates. I'm actually the first and only investigator here at UVA who does have permission to scan babies for research purposes. And so what we've found, so we were administering the same touch paradigm that I described, where we have a piece of plastic or stroke directly on the skin. And when we look at the social relative to the non-social touch, it's kind of just a proof of concept. We are seeing bilateral sensory, primary sensory cortex activation. So even though these babies are sleeping, we need them to stay as still as possible. So the best way is for them to sleep. They are still processing our stimuli, which is really exciting. So we're able to get some functional tasks. And then we're also really interested in understanding how their networks are being set up. So particularly with the idea that having an optimal amount of noise early in life can really shape how these neural networks are being set up for optimal processing. We've been working with some folks in our data science school to help us develop metrics to really pull out white matter tracks from neonates, which is can sometimes be challenging because their contrast in a T1 is just not the same as an adult. It's much more gray matter. So we've been able to come up with some really cool metrics to be able to assess white matter tractology in our neonates using diffusion kurtosis imaging. So that's a future direction that I'm really excited to continue to pursue. Let's see. And finally, so of course, the title of this talk was towards an earlier detection of autism. So we're really hoping that these biomarkers will serve as early markers for autism detection. So we're hoping to be able to essentially create social brain development growth charts. So all babies follow well visits and they are able to see, how are they developing as far as their weight and their height on track. But we have no way to know what these babies are doing socially until about age 2 is when we typically can start reliably diagnosing autism, which there's so much brain development that goes on in those first 2 years of life. So really trying to understand what is the trajectory of these markers so that we can start to pinpoint what babies are diverging from an ideal trajectory. And then also, given the remarkable specificity that I've been showing you, what we're hoping as well is that we can start to stratify these different biotypes of autism. So autism is a highly heterogeneous, very, very variable across the population, but it's likely autisms. So can we start to parse and see maybe what type of autism does someone have and what intervention would be most optimal for any particular individual? So really trying to take a precision medicine approach. So as I said, we're really just aging into our follow-up visit now, where we're able to actually see the outcomes of these babies. So we've only had 7 full-term and 3 preterm babies come back so far. We just started 2 years ago, so just starting to get into this data now. But just as a quick little look and kind of sanity check, we are seeing our preterm babies are scoring lower on our Bailey scales than our full-term babies, as we would expect. And then we've gotten some autistic traits. So all of our full-term babies are screened by the Q-chat. We're seeing a nice variability. We do have a few babies who are scoring within the range that we would bring them in for our diagnostic testing as well. And then we're seeing some variability. So we've had 2 babies so far from our NICU group undergo autism diagnostic testing. And we're seeing 1 baby that's scoring very low and not at all a concern, and 1 baby who is scoring quite high. So we are expecting to see some nice variability and be able to hopefully really parse apart these individual differences as we go on and collect this last wave of our data. In conclusion, just to remind you what I've showed you today, so I've talked about how brain signal entropy is hopefully a promising biomarker of neurodevelopment. So it's aberrant following preterm birth. We tend to see lower levels of brain signal entropy in our preemies. It is showing us really high specificity, so hopefully will be a great biomarker for stratification. And it also is aligning with sensory system maturation. So there's some really interesting neurodevelopmental trajectories that we can hopefully parse apart with this. And finally, what our ultimate goal is to identify the utility of this marker as an early emerging biomarker for autism so that we can intervene while these babies' brains are maximally plastic and ideally optimize their outcomes. So this work, of course, couldn't be done without all of the participating families. And I've also got a wonderful team of trainees who have been absolutely instrumental in helping to collect all this data. And let's see, I guess I went kind of right up to time here, but I'm happy to take any questions if folks are able to stick around a little bit longer. And thank you for your attention.
Thank you, Megan. That was a lot. Yes, you went to the hour, but I think you had so much data. It was warranted. So we appreciate that. I opened this by talking about the fact that you use really integrative approaches, and you showed a lot of those here, which is really exciting. I'm going to open it up to questions, so people can please feel free to raise your virtual hand if you do have a question. I see Ariane already.
Yes.
is ready to
Well,
go. So go for it.
yes, yes, yeah. Well, thank you very much for a really great presentation. I really enjoyed it. I think you did a great job putting it all together, such, big body of work. I just have a question about your metric. I hope it doesn't come across as like combative, but I think when you talk about entropy, as you kind of showed the EEG, the both ends, like really high and really low, are not informative. And there's a sweet spot that like supposedly, kind of mid-range entropy is like where the EEG signal reside in, right? But in passing, when you're talking about your work, you kind of like said, oh, high entropy is good, right? But it's relative to where you're starting. So I think this is kind of like the genesis of the whole idea of information integration theory and using mutual information and transfer entropy to kind of model or like measure or like quantify the state of consciousness or your state of where you are in terms of cognitive state. So because entropy has that limitation. So what are your thoughts on the fact that, I mean, is that your statement that high entropy is a good thing, and therefore is that a good sensitive biomarker? Because my argument is it's sensitive to which direction you're going, high or low entropy. What are your thoughts on this? Thank you.
Yeah, absolutely. Yeah, thank you for that question. So just as, when we think about growth charts, being overweight or being underweight is not ideal. I think there is a sweet spot. So what we're finding, at least in the preterm babies that we've collected so far, is it seems that they're showing lower levels if you consider a neurotypically developing baby the optimal level. There has been a fair amount of work looking at entropy in autism, and some show higher levels relative to neurotypical, some show lower levels relative to neurotypical. So I think it's really important to consider, the task demands, the population. So I think it's a really complex question, but I would certainly expect that there is certainly such a thing as too much noise as well. So far in our work, we're not seeing preterm birth, at least this early in development, emerging as a higher entropy being problematic. But that's why it's really important, I think, to also link to specific behavioral outcomes. So we can see, not only how is the brain responding, but how is it actually predicting what the behavioral outcomes are. So yes, in the cases of what the data I presented, assuming a neurotypical baby is developing as we'd expect and having optimal behavioral outcomes, we're seeing that our preterm babies are having lower levels, less optimal levels in this case.
Thank you very much.
Thank you.
Thanks, Ariane. Robin, go ahead.
Yeah, I have a question is kind of related to Ariane. So I, for my dissertation work, had looked at multi-scale entropy in the EEG signal in adults with autism, and found that they had higher kind of overall entropy than neurotypical adults. And, I kind of turned to the literature on expertise development and how that affects multi-scale entropy in the EEG signal, and effectively, like, as people become more expert in whatever domain it is, playing piano or looking at like diagnosing cancer in a imaging or whatever, the entropy of their neural signals decreases. And the hypothesis is that it's kind of freeing up resources, your brain is becoming more efficient. And I'm wondering if you were to look like further out in development, and as neural pruning takes place, and as like social skills are kind of becoming more habitual, if you will, more natural in the course of development, if you would see kind of a shift in the balance of entropy, where these kids who are born preterm or who are more at risk of an autism diagnosis end up kind of changing places with neurotypical kids, just because of how the neural systems are operating.
Yeah, that's a great question. I think that's why it's so important to ideally be able to do simultaneous EEG and MRI, or at least collect, if not simultaneously, both data streams in the same individual, so that we can really understand how are these signals in the EEG reflecting changes in neural architecture. I also have some animal collaborators where you can obviously do a lot more invasive kind of work. So really excited to be able to explore and manipulate in ways that we can't with our humans how different structural changes or functional or connectivity changes might be reflected then in changes in entropy. So I think we really do need to take a cross-species approach, certainly in a longer longitudinal. The one adult study that I did show today, we did see higher levels were associated with better behavioral outcomes. But again, that's within a very specific, sensory kind of matching domain. So yeah, there's so much more work to be done here, and we don't really understand exactly what the mechanism for entropy is. So that's something that I'm really interested in exploring further with some animal collaborators in particular, and also with the neuroimaging. So certainly, yes, I think that's a very valid question, and something that just as a field we can't answer yet. So ripe for exploration.
Well, I look forward to reading your findings on those studies.
Thank you.
And thanks a lot, Robin. Megan, I'm just going to be greedy for just a moment. I know we're over time, and so people need to drop off. I understand, and we will free you up in just a moment. I just want to ask one more question, though. I'm just thinking about the overall conceptualization and kind of piggybacks a little bit onto both of the previous questions. I think the measurement that the fine-grained entropic measurements, entropy measurements, seem really powerful in terms of understanding brain development, especially as you're describing it in infants and preterm infants. You've conceptualized this, I think, from a larger level, as providing potential traction for identifying early biomarkers of autism or autism subtypes, biotypes. I'm wondering, you're doing a lot of sensory work, and you're kind of getting into this social system through sensory touch and sensory processes. I'm wondering just how specific you think these markers could be for autism as opposed to just neurodiversity more broadly, or even neurodevelopmental delay. So how are you conceptualizing that, and how specific these are versus instead markers of just neurodiversity or neuro risk?
Mm-hmm. That's a great question. So I think it has certainly utility beyond just autism. But what we're hoping to be able to do is link these early sensory experiences with specific autism behaviors. So also really trying to understand the specificity of, autism presents very, very differently. So across every single person, right? So, is there maybe a baby who is having more language-based problems, and maybe they were having more challenges with processing the auditory stimuli very early in life. And of course, there are language issues that exist outside of autism, other language disorders. So can we really just try to predict maybe which babies would benefit from which interventions at what time, and then what type of behavioral phenotype we can expect for them to have, whether it's autistic related or not. We've been focusing a lot on social stimuli to really kind of get at the core deficit within autism, but I think it has utility way beyond just autism as well. So we can really predict just what babies are on a divergent trajectory in general.
It's very cool. It's very hard work to do, I know, but really important mechanistic work. So I want to thank you for both your talk and for letting us ask questions 10 minutes past the hour, and thank everyone for joining.
For sticking around.
Absolutely, that was wonderful, Megan. And thank you for doing it. Yep.
Thank you so much for having me. Appreciate
Absolutely.
it.
And Megan, if you want to just stay on for 2 minutes, I'll
Sure.
hang with you and just touch base real quick.
Great. Thank you.